Hello everyone, welcome to the last lecture of the physiology section, plant growth and development. In this lecture, we will know what is the meaning of growth and what factors influencing growth. In plants, plant can grow into a very big structure like the pumpkins in this picture. It's actually I think it's at least like 100 kilogram. So in biology, the growth meaning is growth means in irreversible increase in mass due to cell division and cell enlargement. This is just simply means whatever is happens it either by more cells or bigger cells. So another term is growth and differentiation. What is differentiation? You can start from a single cells. All living organisms begins as a single cells and increase in mass through cell division and enlargement until a body consisting of possibly billions of cells is formed. Once cells have enlarged, differentiation occurs. That is, cells develop different forms adapted to specific functions such as conduction, support, photosynthesis, storage, and secretion of special substances. If differentiation did not occur, the results would be a shapeless blob with no distinct tissues and little, if any, coordination. The coordination of growth and differentiation of a single cells into tissues, organ, and whole organism is called development. And we can divide the plant life cycle into 4 stages. The first one is embryogenesis, starting from the zygote into the plant embryo, the rudimentary structure of plant organs. And then from embryogenesis, plants grow into an adult. During this stage, we call vegetative development. And then when the plant grow big and develop enough, they will produce flowers. And this stage we call reproductive development. After the plant complete the reproductive development, they will become senescent or they're getting old and die eventually. So what is controlling the development? How do plants know when and where to differentiate it? So the answer to these questions lie in the organism genes and its environment. In 1950s, Stewart and his colleague at Cornell University illustrated this concept with a tissue culture experiment. They began their experiment by isolating cells, parenchyma cells, in carrot tissues and and put them in a nutrient solution. The cells began to divide, grew, and developed into embryo. The embryo eventually grew into a new carrot plant, demonstrating that a cell has all genes necessary to form a new individual. Gene, which is the basic unit of heredity, controlling the synthesis and development of enzymes, which catalyze every metabolic step within cells. Certain genes are programmed to synthesize specific enzymes continually in every living cells, while other genes catalyze the production of enzymes only in specific tissue or at specific times during a plant life cycle. The environment determines which of those genes will be expressed. In an organ or an organism, the environment includes both external and internal factors. The internal environment includes nutrients, vitamins, and hormones. The external environment includes water, minerals, gases, light, and temperature, among many factors. Like I said, gene is the basic unit or the original of everything, and it can be stimulated with external input or external stimuli. For example, plants can perceive the nutrient, soil structure, availability of water. Other organisms like pathogen, symbiotic bacteria, fungi, parasite, neighboring plants, herbivore, or animals that eat them, and they also can perceive gravity, temperature, light, wind, and carbon dioxide, and also oxygen. So the internal input, like I mentioned earlier, it is like photosynthesis status, that developmental stage, health, and so on. Plant will then be determine the pattern of the cells and tissues and organ, while long range of what has long whatever the gene, once it got stimulated, it will produce enzyme, which is protein, and this enzyme will control what the plant will make in metabolic pathway. And finally, these molecules will help the plant to differentiate their cells, and that lead to development. So how the plants respond to environment? As we said earlier, there's both light, temperature, chemical properties like water, nutrients. Plants talking about light, plant didn't only receive the light intensity, it can also perceive how long the light they receive during the day, or they can perceive the season by checking whether how long the light during the day can be. Like in the winter, there's limited amount of time during the day. The day gets shorter. During the summer, the day gets longer, for example. Signal can be like chemical and electrical as well. Electrical is more like a membrane potential. And this signal can be perceived by receptor protein. And receptor protein usually are present in the membrane of the plant, in the plasma membrane, like this one. The signal is in the red dot, and receptor protein can bind to external signal. Once it bound to a signal, the receptor will change their structure. For example, it can change into different shape, and then this signal can be sent to internal signal molecules that can carry the message from one molecule to another, and that will trigger changes in biochemistry and cell shape as well. The signal can be transported into the nucleus in order to control what gene the plant cell will express, the which plant gene it can be suppressed. Think about the central dogma. Gene can make RNA, and RNA can make protein. Protein is like a worker in all the cells. in many metabolic pathway, and we have at least one copy of the gene, and how the cells control how many protein they want to make. They control that by controlling the amount of RNA that can be transferred and make protein. By controlling the RNA, they receive the signal from external stimuli. Once the signal gets into the nucleus, this signal will tell whether they want this gene to have more RNA or not. If the gene makes more RNA will make more protein or make more enzymes that will produce more of some chemicals than the others, for example. So, as we talk about external factor, what are internal factors? For example, we have nutrients, vitamin and hormone. Nutrients are just that substances, elements, and energy for organic molecules that are the building blocks by which an organism develops. We can categorize mineral nutrients into two groups: macronutrients, the plant need in large amount; micronutrient, plant need in smaller amount, but they are essential. Vitamins are coenzymes or part of coenzymes, function as electron donor or electron acceptor, like NADH, NADPH, FADH2, as you have learned in photosynthesis and respiration. And finally, hormones that influence many developmental phenomena. Synthesis of hormones governed by genes. It is usually produced in activating growing cells and transported to another part of the plants where they have specific effects. In this lecture, you will learn about auxin, gibberellin, cytokinins, abscisic acid, and ethylene. But there are a few more that we will not have a chance to learn in this lecture. They are racinosteroids, strigolactones, jasmonic acid, salicylic acid, and spermidine. From this In this video, I will stop right here and we'll come back with some other hormones and probably different processes in plant development.